Hi, my name is Kay Jann. I'm at the USC Stevens Neuroimaging and Informatics Institute at the Keck School of Medicine at USC. And I'm going to present my findings on the association of fMRI complexity with tau PET and cognitive decline in Alzheimer's disease. Disclosure, I have nothing to disclose. Alzheimer's disease is pathologically characterized by the aggregation of extracellular amyloid beta plaques and intracellular tau neurofibrillary tangles. And clinical pathological studies suggest that tauopathy is closer associated with cognitive decline than amyloid beta pathology. These histopathological findings have been recently confirmed also using PET imaging, where, for example, in autosomal dominant Alzheimer's disease, non-carriers, asymptomatic and symptomatic carriers have been compared using amyloid beta or tau PET. We see non-carriers and asymptomatic carriers show already a difference in amyloid deposition. So even if you have no cognitive symptoms, you already show us amyloid deposition, while tau only shows up for symptomatic deposition. So tau seems to be more closely related to the neuronal injury that is related to cognitive decline. More importantly, the tau deposition is specific to the cognitive domain where you have impaired function. So for episodic memory, language, semantic memory, or executive functions, the areas where we find tau deposition seems to be uniquely distinct. Quick background summary. So histopathological and PET imaging show that tau is indicated to be more closely related with cognitive symptoms in Alzheimer's disease than amyloid beta, and the tau is related to decreased neuronal function due to neuronal injury, be that metabolism or gray matter loss. There's one problem with PET, with FDG, amyloid, or tau PET. They require injection of radioactive tracers, and PET scanners as well as radioactive tracers are not as widely available and also more expensive than MRI. So to find an MR-based marker for cognitive function associated with tau-related neuronal injury would be of great interest. I would like to introduce you to a concept here, complexity of fMRI signals as compared to the more well-known metric of functional connectivity. Every voxel or every region of interest shows signal fluctuations over the time course of an fMRI acquisition, while functional connectivity measures the signal similarity between two given areas. Entropy measures the signal complexity within an area. So how complex is the signal behavior of the area, which is a complementary metric to the functional connectivity that's more well-known studied. It has been shown that complexity, as measured by entropy, is reduced with increasing age, and specifically at lower temporal scale, so higher scales of the entropy. We see that for young adults and aged adults, the complexity is reduced at higher scales of entropy. We see that there's an associated with cognitive decline for normal cognitive over early and late MCI to Alzheimer's disease. Again, in higher scales, lower frequency, we find that Alzheimer's disease and late MCI are significantly different from cognitive normal, showing reduced entropy in global gray matter. Also, if you look at regional changes for a default mode network, we find that with reduced entropy in default mode network areas, we show lower MMSE scores. If you go down to one single area, for example, right hippocampus and the Boston naming task as a measure for executive function, we see again reduced multi-scale entropy in the hippocampus associated with reduced cognitive performance in the Boston naming task. How do we calculate entropy? Just very quickly, we need a pattern matching length M, so how long are the patterns that we try to find, and the pattern matching threshold, what do we count as a matching pattern? And for every time point, we count how many patterns we detect at length M and at length M+1. So all the patterns we find for M, and then all the patterns we find for M+1 within this pattern matching threshold. The sample entropy, which is a metric that is well adapted for short noisy signals, is defined as the negative logarithm of the pattern count that we found at m+1 divided by the pattern count at m given the threshold r. The multi-scale entropy extends this sample entropy concept to different frequency scales. So scale 1 is the actual original temporal resolution of the time series. Scale 2, we average 2 consecutive time points into a new time point. So every 2 consecutive time points are producing a new time point. So the time series gets twice as long. For scale 3, time series is a third as long because we match or we calculate a new time point for every 3 consecutive points. And there's a frequency relation between scale and the temporal resolution of the signal that we measure. So the frequency of a given scale is 1 divided by scale times TR. So increasing scales, lower frequencies. The hypothesis for this work is that the complexity of both signals provides an index of the information processing capacity of regional neuronal population and thus is sensitive to tau-related neuronal injury and cognitive decline in the Alzheimer's disease processes. We studied 50 randomly selected subjects from the AAIC2020 cohort, mean age 72.4 years, plus minus 8.2 years, 19 male, 31 females, 25 cognitive normal, 19 MCI, and 6 AD subjects. We parcellated the brain into approximately 200 ROis based on the Craddock atlas. We calculated the complexity as 6 multi-scale entropy scales, and also the tau PET for each of these ROIs, reference versus the cerebellum. Then we calculated Spearman correlations between fMRI multi-scale entropy and tau PET SUVR and control for age, gender, and regional gray matter volume. Here, exemplary maps for tau and entropy in a selective, in representative healthy control and an Alzheimer's disease subject. Tau PET, not much deposition for the healthy control. Entropy, nice gray matter white matter contrast with high complexity entropy in the gray matter. And for AD patients, we see high tau deposition into PCC and hippocampal areas, and we see markedly reduced multi-scale entropy in the PCC and parietal area, and also some in the anterior cingulate cortex. For correlation results, we found significant negative correlation between tau PET and multi-scale entropy, specifically in the precuneus, PCC, and hippocampal temporal areas. So the higher the tau deposition, the lower the multi-scale entropy in these areas. And then we went on and used a random forest classification with 1,000 decision trees and tenfold cross-validation for non-biased classification, where we fed in the functional entropy maps at different pattern matching thresholds, as well as the PET images to see if we can classify cognitively impaired subjects and cognitive normal subjects. The results of this classifier shows that for PET, we get sensitivity, specificity, and accuracy of around 80%. For multi-scale entropy, we can classify MCI, AD, and healthy controls with similar performance, and specifically for MSE at the pattern matching threshold of 0.5, we actually get improved classification performance. Now, what regions of the brain are most informative for this classification? In red is the areas that are most important for the tau PET. In blue, the ones from the multi-scale entropy maps, and the purple is the actual overlap. So classification of PET and MSE is based on the same regions, again, parietal, temporal areas, and that bilaterally. To sum this up, we found correlation classification results for MSE in relation with tau PET revealed areas in the PCC, temporal, and parietal lobes, and these areas have been known to be implicated in tau pathology and cognitive decline. Hence, our overlapping findings support the hypothesis that complexity of resting state fMRI is associated with regional tau protein accumulation measured by PET and might provide a potential marker for information processing capacity of regional neuronal population and prediction of cognitive decline in Alzheimer's disease. I would like to thank my collaborators, the AAIC team, and our funding sources, and you for your attention. Thank you very much.